Hello again, everyone, and welcome to another episode of Creepy Crawly Conversations. I'm Jack Elphinstone, and I'm here with my good friend and Co-Insight Week organizer, Liana Fernandez. And today we are also joined by the brilliant Dr. Jerry Sternberg, a parasitologist and lecturer from the University of Aberdeen. Thank you very much for being with us today, Jerry. I'm really excited to hear what you have to say.
I'm very, very glad to be here too with you, even not physically in the same place.
It's so great to see you face to face. So thank you so much. So just to begin, the first question we're wondering about is your research is primarily concerned with human African trypanosomiasis. And could you please exactly explain for us what exactly this is?
Yes, okay. Well, you may have heard of this as a disease that is more commonly known as sleeping sickness, and it's a disease caused by a parasitic organism. We call them protozoans, single-cell microorganisms, and it's a vector-borne disease. It's transmitted by an insect vector, in this case the tsetse fly, and it's found in sub-Saharan Africa, primarily. The human African trypanosomiasis is only found in sub-Saharan Africa. The parasite gets injected by the flies, which take blood meals. They're blood-feeding insects. They're quite voracious blood feeders. And the parasites then develop in the blood in this very similar way to what people might be familiar with parasites such as malaria parasite, which is obviously much more well-known. Parasites develop in the blood. They make you sick with a fever-like illness, with all sorts of other nasty complications. And the real problem with this parasite is that it can get into your brain. And when it gets into your brain, it causes an invariably fatal encephalitis, and people die. So that's human African trypanosomiasis.
Well, it sounds like quite a nasty disease. You've briefly mentioned that it's vector transmitted. So what exactly does it mean to be a vector? And is the tsetse fly the main vector for human African trypanosomiasis?
The tsetse fly is the only vector, in fact, for human African trypanosomiasis. So what does vector transmission mean? Well, it really means that the fly carries the disease around and injects it into people who are hosts. And more familiar, the vector-borne transmission of diseases is quite common, and insects often are disease vectors, but they're not the only types of disease vectors. So the one, again, that probably most people are familiar with is something like malaria, where the mosquito acts as a vector, and of course you'll know that mosquitoes act as vectors for all sorts of nasty diseases. If we bring that back to a more local context, like in the UK, where we don't have vector-transmitted diseases carried by flies of humans, we do have vector-transmitted disease carried by other arthropods such as ticks, and you'll probably have heard of Lyme disease, for example, is carried by ticks, and that's another form of vector-borne disease because the tick injects the pathogen, in this case, into the person. But obviously we're talking about tsetse flies here now, so yeah, they're called vectors because they carry around the parasite, and they from one place to another, so they're quite mobile. The most brilliant example of a vector actually is another vector of trypanosomiasis, and that's in South America, vampire bats can act as vectors for a form of trypanosomiasis found in cattle. So there you've got one mammal acting as a vector for a disease. So they don't have to be arthropods, invertebrates to be vectors.
That's really interesting. I was also wondering, are tsetse flies found throughout the entirety of Africa, or are they restricted to certain areas? Are they found anywhere else in the world at all?
Right, so they're found really south of the Sahara, and the tsetse belts stretch down to, well, I have a little map, I have a little slide. Shall I share one of my slides with you, because it'll show you exactly what it's found. It's a multi-panel slide, so it's got various other elements on it, but see how my screen sharing works here. So I don't know if you can see that or not, but in the top right-hand panel there's a map of the continent of Africa, and the red splodges there are the areas where we find tsetse flies. And in fact, the top left picture there is a picture of a tsetse fly taking a blood meal. You can see its proboscis inserted into the skin. It saws away with a rasping motion to get into the skin, and it's what's known as a pool blood feeder. So basically what it does is, and they're quite painful. I've been bitten lots of times, and normally during a normal insect week, I would actually have some tsetse flies and I would demonstrate them feeding on me. You know when they're feeding, and they basically rasp away, and they break lots of capillaries and create a little pool of blood at the tip of where the proboscis is in the skin, and they suck away from that. And then just to tell you what the other pictures in that panel are, the lower left picture is specimens from David Livingstone's collection, because David Livingstone did a lot of interesting field biology amongst his other work while he was traveling in southern Africa in the 19th century, and he has some specimens of tsetse flies there. And I won't talk about the other panel there. That's a bit of microscopy of the salivary glands of a fly, and that's kind of, maybe we'll come back to that later. So that's that. I'm going to just stop that screen share. Hopefully you're back to me now.
Yep.
So that's the distribution of flies. However, just to carry on from your question, although that's the distribution of tsetse flies now, today, we know that if we go back in evolutionary time, probably we need to go back a couple of million years and we'll have found tsetse flies more widely distributed around the world. But presently, they're restricted to sub-Saharan Africa.
I suppose it's quite a relief that these flies have a restricted distribution, because I don't fancy coming across one. But knowing that these flies help transmit the trypanosomes, can you explain exactly how that occurs? What do the flies do to aid transmission of these trypanosomes?
So there's a life cycle of the trypanosome. So in the fly, the trypanosome parasites, these microscopic parasites, develop in the salivary glands of the fly. So the flies have got these glands where they produce saliva, and the parasites develop in those salivary glands. When the flies, and this applies to most blood-feeding insects, that when they take blood meals from people, and it's not just people, by the way, the tsetse flies are a problem for. They're a huge problem for livestock. So they don't just feed on humans; they feed on a wide range of mammals. But when they feed on us, flies have got some big challenges. All blood-feeding insects have got some big challenges. We have our own immune defenses that come into play. That, we don't really like being bitten by blood-feeding insects. And so flies have to overcome all the defenses we have against things, basically other animals pricking little needle-like proboscises into us and trying to suck our blood. So they salivate, and the saliva's got some important functions in allowing them to take blood meals. So the saliva contains all sorts of cocktails of chemicals that interfere with our defense processes against infection. And so that's why the flies salivate, and they salivate a tiny volume of saliva. When a fly, like the fly in the photograph I just shared, takes a blood meal, it salivates about 1 microliter of saliva. That's a millionth of a liter. So that's the tiniest drop of fluid you could put. If you could imagine the tiniest drop of fluid you could see on the end of a teaspoon, that would be a microliter. That microliter of saliva will carry some parasites in the salivary gland. You just need 100 parasites in that microliter of saliva, and then they will start to develop inside the human body or the other host, the other mammal hosts. And then they go through a series of stages of development. They get into the bloodstream, they invade various tissues, and eventually they could get into the brain. But it's a cycle. So for the disease transmission to be sustained, another fly has to come along at a later time point and suck some blood up and become infected, thus perpetuating the transmission cycle. So that's really how this blood feeding transmits the disease.
So yeah, these flies are obviously very central to the transmission of the disease. And if we know that these flies are responsible for helping spread the disease, then can we control it by controlling tsetse fly populations? If this is possible, and if so, what methods can be used to help regulate tsetse fly populations?
Yeah, well, quite a few actually, and so the tsetse populations can be controlled. Well, there are a number of ways that we control that they're controlled today. Put it this way: in areas where flies are controlled, we can control habitat to make the habitat less hospitable for flies, first of all. And in fact, certainly, it's now thought that the distribution of the diseases carried by tsetse flies really only spread widely in the eastern and southern Africa relatively recently, in fact, in the late 19th century. And this is a very interesting kind of bit of, I suppose, history of disease and anthropology, because in the late 19th century, during the colonial period, one of the things that happened, not intentionally, was through the introduction of livestock from Asia and Europe, a viral disease called rinderpest, which you might have heard of, got introduced into southern and eastern Africa. And it wiped out indigenous livestock, wiped them out, and it basically drove lots of the pastoralists who did small-scale pastoral farming on the savannas of eastern and southern Africa out of existence. And that savanna then changed from grassland to the kind of environment that we now see when we go to the sort of iconic game reserves like the Serengeti, which are, there is grassland, there's a lot of bush, small stands of forest, lots of acacia bush and stuff like that. And that's ideal habitat for tsetse flies. And that's when the tsetse flies moved in. So habitat control can actually do a lot for managing the risks of flies, because if you make the habitat less hospitable for the resting flies, you won't eliminate them, but you'll have less interaction with them. The second thing you can do with the flies is, and this is practiced quite widely in some areas, particularly I believe in Botswana, insecticide treatment to control the flies. Now, it's worth mentioning that all this effort to fly control has got nothing to do with eliminating human African trypanosomiasis, the disease. This is all to do with eliminating the threat of this disease to cattle. And because it's a big deal. And in fact, the cost in terms of the economic cost, and also the cost in terms of human development of the livestock disease, is much greater than that of the human disease. And so there's various now quite refined techniques of insecticide application that are used to manage flies, particularly in the ranching areas of countries like Botswana. And then on a more local level, there's various ways you can manage local exposure using traps and targets. I've got a couple of pictures here. I don't know if this is any. Let's just see what happens. Just to show you what I mean by that, let me just share the screen again. So you might recognize this set of images. I use them quite a lot in lectures, but they're on the screen now. You've got two different types of traps and targets. The top image is a target, which is just a sheet of, it's an old sack basically that's been opened up, been painted with that characteristic color, because that trap flies, the tsetse flies have got some very interesting, they're very clever actually, and they've got some very, very cute visually. And they're strongly attracted to blue, and they're very strongly attracted to these linear contrasting backgrounds. And so what you do with the target is you basically arrange that every 3 months that is sprayed with a persistent pyrethroid insecticide, and then when the flies land on it, they pick up the insecticide and die. And then the lower image is known as a pyramidal trap, and that's a little bit more complicated to make. It requires a bit more sewing, but again, the blue color attracts the flies, and then within the trap there's a kind of a maze. The flies go into the maze and they can't get out again. So you can control in very local areas, you can control flies that way, using traps and targets. And then the other way we can control flies is through a technique called the sterile insect technique, which you might have heard of. That's probably a more limited use, I think now. It was used to eliminate the tsetse fly from one island, that of Zanzibar, in the 1980s, and it involves releasing sterile male flies into the environment. And the sterile flies, so tsetse fly, basically females mate once, and if they mate with a sterile fly, then basically basically none of their eggs develop. So basically you can suppress fly populations using sterile insect technique. But it really only works, it's got two problems associated with it. You first of all have to get thousands and thousands of sterile male flies to release. And that involves irradiation, and that's quite complicated to organize. And interestingly enough, that's the reason why the largest tsetse fly colony for generating flies for research and other purposes is not in Africa, it's in Slovakia, in Bratislava. And historically, the reason for that is that originally it was in Vienna, actually, and it was associated with the International Atomic Energy Authority, who obviously one of their functions is to look for peaceful or uses for nuclear technology that are not to do with making bombs, basically. And one of those uses is to irradiate flies and other insects with sterile insect technique. But there's a logistic problem in getting all those sterile male flies, and also it really only works if you have an environment where there's no risk of reinvasion. So an island is ideal, but it's really not applicable, I don't think, on a wider scale.
I'm glad that it's not all doom and gloom and that there is some potential to control this disease as well as these flies. But I'm interested, in addition to your work on human African trypanosomiasis, you've also carried out research on American trypanosomiasis, which is otherwise known as Chagas disease. So is this disease also spread by tsetse flies, or are there other vectors involved?
So vector's completely different for South American trypanosomiasis. Interesting enough, though, just before the trypanosomiasis that African trypanosomiasis is restricted to Africa, as you might imagine, but there's various animal trypanosomes that are transmitted by tsetse flies in sub-Saharan Africa that are transmitted by other insects in South America. And as I mentioned as well, there's the ones transmitted by vampire bats. But those are animal diseases. Chagas disease, the South American trypanosomiasis, is a completely different disease to African trypanosomiasis. They're both the parasites are both called trypanosomes. They are related, but they're very different in their life cycles and their cell biology, if you like. And the vectors are very different. So tsetse flies, only found in Africa. They're remarkable flies, tsetse flies, because they're fascinating flies. They're really, really unique. Almost the insects that transmit South American trypanosomiasis are Hemiptera. They're much more widespread taxon, and they're bugs basically, and they're known. They've got lots of names. Some people call them vampire bugs. Charles Darwin wrote about them and described them as vampire bugs, I think, in The Voyage of the Beagle. Some people think that Darwin died of Chagas disease. I think it's a bit of an urban legend, but nobody will ever know because the family won't allow the body to be disinterred to do PCR testing now. We can find that out. So they're sometimes known as vampire bugs. They're sometimes known as kissing bugs. They're sometimes known as rejuveate bugs. Yes, so they're completely different animal, and they've got a slightly different mode of transmission. If I just flash up an image of one of the species of kissing bug, you will hopefully see this. Let's just. So I'm sorry, I'll do another screen share here. Okay, just briefly, well, sorry, there's some statistics there as well, but the image there is, this is Trypanosomiasis infestans, and it's about to feed on somebody. This isn't me. We had them in the lab, but they've not survived lockdown, unfortunately. Well, I don't think so, because they should be fed every few weeks. These bugs, they're about, that bug that is in the image there is about 1 centimetre, maybe 50 millimetres in length. And they don't infect people by their saliva, though. They become infected through sucking up blood from infected people, but they infect other people because they've got very bad toilet habits. And basically, while they're taking blood, while they're blood feeding, at the same time they defecate. So they're dropping basically poop on the skin around the bite site. And some of that can get into the bite wound, or otherwise it can get rubbed into people's eyes and things like that. And that's how people become infected with the parasite that causes Chagas disease. And as you can see, this is a serious health problem. And the numbers on there, I put them on there because I forget these numbers all the time, but they reckon that 8 million people are infected. And there's a lot of deaths per year. It's emergent in Europe and USA. This is a little old statistic. They reckon 250,000 people infected in the USA now, but it's important to bear in mind, I'm just going to stop the screen share now, but it's important to bear in mind that 250,000 people who might be infected in the United States, they're not being infected by bugs in the United States. They're traveled to the United States carrying the infection, because the disease is a very chronic disease. So you can carry this parasite around in your body for many, many years before you develop any serious health complications. One of the interesting things about Chagas disease, though, is that the bugs are not just restricted to South America. So these Trypanosomiasis, these rejuvenated bugs are found throughout the Americas. So they stretch as far, the distribution of the bug stretches us up to, in the United States, up to the Midwestern states of the USA. And yet, the incidence of infection, there's a term you might have heard of, it's a horrible one of these horrible medical terms called autochthonous transmission. It just means that if you get Chagas disease, if you're one of the people, thousands of people who have Chagas disease in the United States because you travel to the United States with the disease, then that's not autochthonous transmission. If you got bitten in the United States by a bug carrying the disease and got infected, that's autochthonous transmission. That's actually very rare. And there's probably only about 30 or 40 cases that have been reported in the USA. And yet we know the bugs have got the parasites in the USA. And that says two things. It says that, but the two reasons for that. One is that, first of all, the species of bug that are found in the United States have got slightly different habits to some of the species in South America, in that their habits mean they don't reside close to human habitation quite so much. And secondly, it's to do with the construction of housing. Because the bugs, we've done a lot of work in Bolivia, for example. If you're in Bolivia, the reason you're in contact with Trypanosomiasis infestans, this bug that transmits the disease, is that it during the day, it hides away in the cracks in the walls of your house. And it comes out at night, a bit like bed bugs do in Europe. You know, the bugs come out at night when you're sleeping and they find you. And if you have a house that's well constructed, doesn't have a thatched roof, doesn't have lots of cracks in the walls, then you won't have bugs in your house. And so there's been great progress actually in controlling these bugs in South America in the last few decades through a program called the Southern Cone Program, where it's a triumph of actually intergovernmental cooperation through health services cooperating, and a lot of it's been based on people's improvement in housing, application of persistent insecticides in people's houses and stuff like that. So there's been a great reduction in transmission of Chagas disease, even though there's still a lot of it about.
So it seems like quite a lot of funds and effort have gone into regulating and controlling these, kissing bug populations. but could a vaccine be developed instead, just as an like easier method? Why has this not occurred yet?
Well, it depends. In the case of the African trypanosomes, it'll never happen, I don't think, because the parasite that causes that disease, the sleeping sickness in Africa, it has this trick, you might have heard of it. It's called, the technical term for its antigenic variation. Basically means that it changes its coat all the time. And our immune system recognizes the coat, but then it changes its coat. So it's generally accepted that we'll never get a vaccine against African trypanosomiasis for that reason. Now, the South American trypanosomes are slightly different, and there have been ongoing efforts to try to develop vaccines against South American trypanosomiasis, and it might yet be possible, but nobody yet has found the right combination of antigens to allow that vaccination to take place.
Quite interesting. So, given that there's maybe not a potential for a vaccine to develop, in your own personal opinion, would you think that complete eradication of both the kissing bugs and tsetse flies be a favourable solution to treat trypanosome infections?
Yeah, okay. Well, that's a very good question, and it's a very difficult one to answer because, it's one that is debated by infectious disease people and bioethicists and conservationists and philosophers. Is it right to eliminate completely a whole species from the planet? And so that you have to strike a balance. In the case of the tsetse fly, actually, I think if it was possible, it might be quite good to completely eliminate the tsetse fly, because we know that it probably isn't pivotal it's probably not an ecologically pivotal species. It doesn't seem to be an essential item in any food chain, for example. Fortunately, the ethical question of the tsetse fly has not really needed to be tested that hard, because the practicality of complete elimination of the tsetse fly is so beyond us at the moment. It's not going to happen. We can control it locally, and that seems to be the best way forward. We can control it by habitat management. But complete elimination seems very unlikely, even though it might be a desirable outcome. It will have some other consequences as well. I think they're probably quite positive consequences for human development, but some ecologists might balk at the prospect of the fact that a lot of the savanna areas of sub-Saharan Africa, which are currently not sustainable for agriculture, would become sustainable for agriculture. So it could put pressure on ecosystems that we value, even though those ecosystems aren't very productive in terms of producing food for a population that needs food. So that's that's Africa. In South America, I think it's even more unlikely that we will ever eliminate this insect. It's impossible to target it specifically enough without huge collateral damage on other invertebrate populations. And its sylvatic habitats are so hard to reach. The forest habitats that it hides away in are so hard to reach. I don't think it's realistically going to happen. I think it's a realistic sort of plausibility. So I don't think we need to approach that question either. I think both of these vectors and their diseases could be better managed by locally managing exposure of people to the vectors so that we can live with them, we don't get exposed to them, and then having improved diagnostics and treatment for the diseases that they cause.
Yeah, no, you briefly mentioned before that Chagas disease has made its way into Europe a little bit, and I have finally one slightly more controversial question, but in your personal opinion, is there any point in providing funding to international projects aiming to treat these diseases when we ourselves aren't affected in the UK? Well, I think the way you need to look at that is the number of angles on that question. I mean, first of all, you could just say, well, why provide any international development aid of any sort at all? Yeah.
So, assuming you are going to do so, then human health, and and agricultural health are fundamental to development and human welfare. So I think that, you know, that's perfectly appropriate that we should be supporting that sort of thing. We pay the price. Obviously, we live in an interconnected world, and so apart from the fact those diseases might never come here, and in fact, of course, as you say, they do come here through travellers bringing them here. Mm-hmm. So we're not free or immune from these infections, and then we also, there's a big cost due to inequalities across the planet due to these impairments to development. And so many of the impairments to development are caused by health problems, neglected tropical diseases, malnutrition, and all of those other things. And so by addressing those, we can make a more equal world, and then we're all better off. So I think that, you know, that question is one, you know, I don't agree that we shouldn't be restricting our support for health problems to just the ones that occur in the UK. And of course, with climate change, who knows what's going to turn up here in the future.
Yeah.
Mm-hmm. Well, I agree with you there. It's all been very thought-provoking, interesting, but sadly that's all we have time for today. But I do just want to give Jerry a massive thank you for joining us today. And if you would like to see more of his research, then we will provide a link at the bottom of the description where you can access some of his publications. But until then, thank you all for listening, and happy Northeast National Insight Week. We'll see you all next time.
Happy Northeast National Insight Week. Thank you.
Yeah, thanks for having me.